Thank you so much. Thank you for that incredibly kind introduction, and also for the opportunity to speak to all of you. I love talking to students. I think this is maybe the most impactful thing that any of us do. So what I'm going to do today is talk about bodies, minds, and environments. I'm going to say some pretty unusual things, I think, and I see these things in a little bit of a different way than they've been handled in the past. But the middle part of the talk is going to be a case study showing how applying these ideas to a model system, the collective intelligence of cells during morphogenesis, leads to very practical applications in biomedicine, bioengineering, and so on. And I think it's important that these kind of conceptual and philosophical things that we develop here are tested in terms of their ability to drive new discovery. So that's what I'll do. Okay. Oh, and if you want to dive into any of the data sets, the software, everything underlying the things I'm telling you is all here. This is our official lab website, and my own personal thoughts about what some of these things mean are at this blog here. So the first thing I want to start with is this classic vision of nature. And so this piece was called Adam Names the Animals in the Garden of Eden. And there are two things to note here. One that I think is very important that we change, and I think it's already starting to change. And then the other one is profound and deep, and we're going to have to pursue it to the end. The first is that what's happening here is that there is a categorical demarcation between natural kinds. So you have here the various species; they're all distinct. Here's Adam; he's distinct, of course, from all of them. And we know what all of these things are, and they're kind of very, very specific standard set of animals with behaviors and form and so on. And so that I think we're going to have to change. And I'm going to, in this talk, run through a kind of a spectrum of progressively weirder kinds of beings. But the thing that I think is profoundly important here is that in this old story, it was up to Adam to name the animals. God couldn't do it. The angels couldn't do it. It had to be Adam that named the animals. And the thing is that in these traditions, naming something means that you've discovered its true inner nature. Either you've found out its true inner nature, or you actually assigned it a name, which means you are a co-creator with it of its inner nature. And we're going to have to do that, I think, for an increasingly diverse set of bodies, minds, and environments. Now, minds are pretty easy to detect in things like brainy mammals. So here's this squirrel-like creature. He's going to set up a little accident scene here to get the attention of his owners, right? So here he's faking he's faking this set of events, and he's putting it on his neck. He knows exactly what gets attention. He's in fact going to look to see if they're watching. It's kind of an amazing thing, and it's easy to recognize. It's easy to recognize this kind of behavior in certain kinds of beings, because we share the same spatial temporal scale in which we operate, in which we deploy intelligence. So we work in the same space. We have similar goals. So we're okay at detecting things like this. But I want to bring our attention to this kind of analogy here, that in this enormous electromagnetic spectrum, prior to having a good theory of what underlies this spectrum, two things were true. First, we were only sensitive to an extremely narrow range of it. So our vision operates here. We were completely oblivious to the existence of all of this stuff, right? And there were some phenomena, magnets and static electricity and lightning and visible light and so on, that we thought were all different kinds of things. We thought they were different, distinct natural kinds. And then when we understand true, and so this is the power of unification. When you really understand what's going on, you find out that it is in fact one spectrum. These are all different entities on the same spectrum. And that kind of a thing allows you to improve your ability to sense and exploit the whole spectrum. In other words, now we have technologies that can operate along this whole thing. So what I'm going to claim is that we are in the same position with respect to minds and recognizing minds. In other words, we have a kind of mind blindness because of our evolutionary history that only allows us to recognize minds and environments. It's the same thing about environments. We only understand this sort of three-dimensional environment really so far, and we're mostly blind to this other stuff. And the things that we have discovered, a lot of people think are distinct. These are not the same thing, and I'm going to argue that it is in fact a spectrum. One of my personal heroes was Alan Turing, and he, of course, was interested in intelligence, in diverse media, in programmability, in computing, and all of that. So he was interested in problem-solving machines, so intelligence through reprogrammability. It's a kind of plasticity. But towards the end of his life, he also wrote this incredible, well-known paper, The Chemical Basis of Morphogenesis. So he was interested in how chemicals self-assemble order in embryonic development. Now, why would somebody who's interested in intelligence and computing and programming and things like that, why would they be looking at chemicals in development? And I think Turing saw a very profound truth that is very important, and that is our first step towards really being able to work across the spectrum of minds, bodies, and environment, which is that our origin as embryos enables us to have some deep insight into what's going on here, because there's a deep symmetry between the self-construction of bodies and minds. I think he saw this, and I think it's absolutely right. The process by which bodies come into the world and self-organize is very, very, very deeply homologous to the process by which minds come into the world. And once we start to understand that, that is the gateway to really being able to detect some very unconventional intelligences that are all around us. So today, I want to do three things. I want to show you a little bit of unconventional biology as inspiration for understanding for enlarging our understanding of embodiment across substrates. I want to show you some specific examples. So this is a progress made in our group in communicating with the agential material of life. We don't have any alien life to deal with yet, but this is the closest thing. We can communicate now with a very different kind of intelligence that shares some similarities with us, but in many ways is very different. And I'm going to show you how we do it using the bioelectric interface to the cellular collective intelligence. And towards the end, I'm going to address this issue of where do novel beings get their goals, because part of our framework, and I don't have time to go into it in too much detail today, but we can, if people want to ask questions about it afterwards, you can. I think one of the central things about being an agent and having intelligence is the scope of your goals, and so that raises the question of where do goals come from, especially in novel systems. So the framework that I'm working on really tries to allow us to build a spectrum and a continuum of very diverse kinds of intelligence. So, okay, the obvious kind of primates and apes and birds and things like that. And then all kinds of weird biologicals, synthetic biology, so engineered new life forms, AI, whether purely software or embodied in some way. Maybe someday exobiological agents, and a couple of even stranger things. I'll briefly mention, if I have time, I'll briefly mention patterns within media as a kind of intelligence. And then there's even one more step that I definitely don't have time to talk about today. And so what I would like to do is to find a framework and to construct a framework in which we can think about all of these things as fundamentally on the same spectrum. I'm not the first person, of course, to try. So here's 1943, Rosenbluth, Wiener, and Bigelow had this kind of spectrum. They were trying to understand what is it that happens when you move from passive matter all the way up through human level metacognition and beyond. And of course, they had a very kind of cybernetic view of this. And I think in many ways, they were on the right track. But this has a much more careful, this paper has a much more careful layout of the things that I'm going to mention. Okay, so let's look at some of this unconventional biology. And I'm going to describe a little bit the unique features of this material. Why does this work in a way that none of our current technologies work? So here are some examples that you may have not seen before, because no one covers this in typical courses, because they don't match the kinds of mechanisms and the kinds of dynamics that are now thought to govern all these processes. So here's an example. This is a tadpole of the frog Xenobot laevis. Here's the mouth. Here are the nostrils, the brain, the tail. This is the gut. This yellow thing is the gut. So this whole thing is the head. And what you'll notice is we've done two things. First of all, we've prevented the primary eyes from forming, but also we put an eye on the tail. And I'll show you later how we do that. So this eye makes, first of all, these cells make an eye despite their weird location. Second, they make an optic nerve. The optic nerve comes out and it does not go to the brain. It synapses either on the spinal cord here, or sometimes on the gut, or sometimes nowhere at all. And the amazing thing is that when we built this device to check the vision of these guys by testing them in visual learning assays, so they learn to stay away from certain light spots and things like that, and they get rewards and punishments. what we find is that they can see perfectly well. These animals can see. Now, it's kind of remarkable and unexpected that when you make a creature with a radically altered sensory motor architecture, okay, where the eyes are not where they're supposed to be, they're not connected to the brain in the way they're supposed to, it works out of the box the first time. You don't need extra rounds of evolutionary mutation, adaptation, selection. You don't need any of that. This works. Why does this work? Where does this plasticity come from? Furthermore, there are other kinds of plasticity. For example, these tadpoles, they have to turn into a frog. And so to do that during normal metamorphosis, they rearrange their face. So the eyes move forward, the nostrils move, the jaws, everything kind of moves. And before, it was thought that, well, this is a hardwired process. Somehow the genome tells each organ which direction to move and how far, and you go from a normal tadpole to a normal frog. We wanted to test this, and this is really critical. When you want to assess the intelligence of a process, you have to do perturbative experiments. You have to put some distance between the system and its goals and see what it can do. So we made these so-called Picasso frogs, where basically what we do is we scramble all the craniofacial organs. So this eye is on top of the head, the mouth is off to the side, everything is just scrambled. And what we found out is that they make pretty normal frogs, because all of these cells and tissues do not move in prescribed hardwired ways. They do whatever they need to do to get to the final outcome. In other words, they go through novel paths. Sometimes they go too far and they actually have to take a step back. So what the genetics actually gives you, and this is really important, is a problem-solving agent. It specifies a system that executes towards a goal state, and it can actually adapt to novel starting conditions and things like this. And this process now, so we're starting to see the importance of this plasticity and the navigation of this anatomical space here. What's really important about this process is that it's reliable. In other words, yeah, acorns, billions and billions of times all over the planet, acorns make exactly this. And so if you focus just on this process, you would be tempted to, as many textbooks say, is that this is what the oak genome encodes. The oak genome tells the cells how to make exactly this, right? But in fact, that isn't what's happening here. There's a bioengineer, a non-human bioengineer, this wasp, and this wasp has a way of prompting the cells. So it puts down some signals, it prompts the cells, and the cells build these incredible structures called galls. The wasp is not building this like a 3D printer, the way that actually the wasp builds its nest. That's not what this is. These are made of plant cells, not wasp cells. And the wasp is basically hacking the morphogenetic competency of these sleep cells. But if this wasn't happening, you would have no idea that this thing that reliably makes this flat green structure can make something like this. So now we see that reliability is really often hides the plasticity and the hackability of competent systems. So we need to really understand how do we start to communicate novel goals to these kinds of systems. One other thing that's really important is that information in biology is treated very differently than it is in current computer science. As we think about how could we copy this kind of plasticity, robustness, we have to really think about how information is handled. And I want to give you a very simple example. So this is a caterpillar. It's a soft-bodied kind of creature, lives in a two-dimensional world, crawls around leaves, and it eats the leaves, and it has this brain suitable for that process. Then it has to turn into this butterfly, which lives in a three-dimensional world. And during metamorphosis, the brain is basically dissolved. Most of the cells are killed off, the connections are broken, it refactors the new brain. Now, the amazing thing is that if you train the caterpillar, and this is reviewed in these papers, if you train the caterpillar, the butterfly remembers the original information. So the first thing you might wonder is, how do you store memories when the medium is being completely torn apart and rebuilt? Okay, and that's a good question. But there's a deeper question here, which is something else. It's not about, this whole process is not about this fidelity of the data. It's not about keeping the information while the material is degraded, because you might think, well, redundancy and error correcting codes, we have ways of dealing with that. But there's something much weirder going on, which is that the actual memories of the caterpillar are of no use to the butterfly. The butterfly doesn't move the way the caterpillar moves. It's a hard-bodied creature. You can't control it the same way that you can a soft body. It doesn't care about leaves. It wants nectar. It drinks nectar, and it flies in a three-dimensional world. The actual memories, for example, seeing a certain stimulus and going and crawling over to eat the leaves, is of it's of absolutely no use to the butterfly. If that information is going to persist, it needs to be remapped. It needs to be reinterpreted by the machinery. And so this issue of having allegiance to the salience of information, not the fidelity of it, but the salience of it, which means taking an active role at all times to reinterpret whatever information you have, is really what biology is all about. So this is in a way, it's called the paradox of change, because any species, so let's just talk about the evolutionary scale for a moment. Any species faces a paradox. If you don't change, if you stay exactly the same, you are pretty much guaranteed to die out eventually. If you do change, then you become something else, and in a certain sense, again, you're gone. The prior you is no longer there. And so what's the solution? And the solution, I think, is to realize that both on the evolutionary scale and on the cognitive scale, we are not objects, and this theme will come back at the end when I talk about patterns as agents. We are not objects. We are self-constructing stories. And what I mean by that is that at any given moment, you as a cognitive being have no access to the past. What you have access to is the memory traces, the engrams that past experience has left for you in your brain and body. So they are messages from your past self, right? At any given moment, you are a selflet. You are one of these slices in this time extended history. And what you have are messages from your past self. Now, messages have to be interpreted. And like any good message passing from lateral interactions, the same thing about vertical interactions through time, you are under no obligation to interpret those messages in exactly the same way that prior you had interpreted them. The information that you have, the molecular engrams that are left in your brain by past experience, every single, whatever, 300 milliseconds or less, you have to actively reconstruct those memories to tell and retell a coherent story that makes sense now, and more importantly, going into the future. So this means that what's happening here is that there's this bottleneck where past experiences are compressed into very, very sparse, very thin representations, such as memory traces. So the now moment is the middle of this bottleneck. And if you do any coding, you recognize this as some sort of autoencoder architecture. And that process can be algorithmic. So compressing prior instances into a generative rule is some kind of an algorithmic process. But this is a creative process because you've lost information here. You don't know actually what these memories have meant. You never know what your memories have meant. You reconstruct what your memories mean. And if things stay the same, then your reconstruction is often very similar to what it was. But it doesn't have to be. It doesn't have to be through childhood and puberty in humans. It doesn't have to be the same in butterflies and caterpillars. And the thing is that this exact same scheme applies on the evolutionary and developmental scale, because what you're handed as an embryo coming into the world, what you're handed is a very compressed representation in the form of an egg and its contents, including DNA and many other maternal determinants. You're handed that information, and now it's up to you to reinterpret it. This is a very different idea than what we're normally taught, which is that you're handed some DNA. The DNA has a fixed meaning. And it drives mechanically. It drives the chemical processes that create your body and your mind. I'm proposing a very different view, which is that what evolution actually makes are problem-solving agents that are given prompts in the form of these memory traces. And then it's whether those be evolutionary or genetic or other kinds. And across scales, the goal of all levels of life is to reinterpret them in whatever way makes sense. Here's an example. This is maybe one of my favorite examples. This is a cross section through a kidney tubule in a newt. And normally there's 8 to 10 cells that work together to do this. Now, one thing you can do is you can force the early cells to have multiple copies of the chromosomes. So you can make polyploid newts that have 3N, 4N, 6N, and whatever. When you do this, the cells get bigger, but you still get a normal newt, and the newt is the same size. So that's amazing. Actually, it's invariant to the amount of genetic material it has, but it stays the same size. And how does it do that? Well, fewer cells, but the cells are bigger. And then when you make truly gigantic cells that way, just one cell will bend around itself and leave a lumen in the middle. Now, this is a completely different molecular mechanism. This is cytoskeletal bending. This is cell-to-cell communication. And that means that what the system is able to do is to use different molecular affordances to solve a problem in this anatomical space. And that is basically the definition of, well, it's one definition of intelligence, is what they test on IQ tests, where they say, you know, solve this problem using these tools that you have, and often you have to be very creative in doing it. Think about what it means when you're a newt coming into the world. You can't count on the external environment, fine, but you also can't count on your own parts. You don't know how many copies of your genetic material you're going to have. You don't know what size your cells are going to be. You don't know how many cells you're going to have. Everything is unreliable. Biology is not doing this with error correcting codes and trying to hold on to the fidelity of information. It is doing it by assuming from the very start that the biological material is unreliable. You cannot avoid changing. Everything is going to change. You have to lean into it. And what evolution has to produce are creative problem-solving agents that interpret that information as best as they can to solve problems. And now you can see why that tadpole with the eye on its tail could see. It's because it never assumed that the eyes were going to be in the correct place to begin with. Usually it is, and then you get the normal default outcome, but it has to solve this problem fresh every single time. It never knows what it's going to encounter, and all of it is a problem-solving kind of thing. So so this is just the same thing. This bow tie, you know, both on the scale of on the cognitive scale and on the developmental scale, that compression here is what drives a kind of intelligence all the way from the beginning, right? That ability to solve novelty is baked in from the beginning. And this is we've got a new paper coming in a couple of weeks that talks about why life doesn't overtrain on these priors. And so what happens next is, because the material is so unreliable, what happens is that evolution, there's basically a kind of feedback loop, a ratchet between intelligence and evolution, because the more unreliable the material is, the better it has to get at interpreting novelty. And the better it is at interpreting and adapting to novel circumstances, the harder it is for evolution to select on the genome, because what selection doesn't see the genome, selection sees the functionality. And that means that evolution does much of its work on the intelligence process, the interpretation process, as opposed to the hardware specification of the genome. And so this is there. We did a lot of simulation work on this. You can see that here. So now you're seeing what's going on here. The living material is importantly different than the kind of passive materials that we work with in, let's say, robotics or computer science, where there is one correct interpretation of the data, and we try to hold on to it at all by having layers of abstraction. We try to hold on to it at all costs. Biology does exactly the opposite. So that starts to help us understand the origin and the kind of the nature of this biology, where at every level of organization there is a kind of intelligence that is reinterpreting information. So what I'm going to do next for probably the next 20 minutes or so is give you an example of an unconventional intelligence that arises from this process. And so let's look at where we come from and what we really are. So all of us took this journey. We started life as a single cell. So this is an unfertilized oocyte. It's a little quiescent ball of chemicals that people would describe as being amenable to the laws of physics and chemistry. And then slowly and gradually, through this amazing process of morphogenesis, we become one of these things, or even something like this. And the important thing to note here is that there is no special point in which you transition from chemistry to being the subject of psychology. This is slow and gradual, which means that we have to understand the scaling of intelligence, not magic categories of chemical machines versus real intelligent beings. Those categories, I think, are not binary at all. And what we have to actually tell are stories of transformation. How did we get here? And by the way, this isn't the end. The collective intelligence that creates this actually can fall apart, and that's what we mean by cancer. And it can reconfigure itself, and I'll show you. Even after the death of the original human, you can get novel life forms out of those cells called Anthrobots, and I'll show you those. So the way that all this works. So how does this work? You know, this is an amazing process. How does it work? Well, the first thing to realize is that we are all made of cells. Cells are extremely competent. This is a single cell. This is a lacrimaria. This is a free living organism, but you get the idea that there's no brain, there's no nervous system. The thing does everything it needs to do on a single cell, on the single cell level. And what they do is they join together to make embryos. Now, what is an embryo? Well, we can look at an early blastoderm. So here are, I don't know, 100,000 or a million cells. Why do we look at that and we say there's one embryo? What is there one of? What are we counting when we look at this blastoderm? Well, what we're counting is alignment toward a very particular story. We're counting intent. We're counting alignment. All of these cells have the same model of where in anatomical space they are going to go. This is where they're going to go. This is why this works. And in fact, if you and I did this with duck embryos as a graduate student, you take a little needle and you put some scratches into this blastoderm. Each one of these islands is going to, not feeling the presence of the others, it's going to self-organize into a new embryo. And so eventually they might heal up or they might not. And that's how you get twins and triplets and whatever. So the number of individuals, the number of beings in this blastoderm is not set. It's not set by the genetics. It can be anywhere from 0, 1 to probably half a dozen. And they spontaneously sort of self-organize, and this is the process that brings new bodies and new minds into the world. And it has many interesting. We can talk about this in the Q&A. It has many interesting similarities to issues in cognition, where again the question of how many beings are in a certain piece of hardware, let's say in a human brain, how many beings are in there? Well, we have split brain patients who have at least two. The left and right hemispheres have different opinions on things. Dissociative identity disorders, where you can have dozens of personalities within the same brain, and so on. So this is a very dynamic process at every scale, because okay, these cellular collectives certainly do some interesting things, but even individual cells can do it because the molecular networks inside of them are already smart. In other words, molecular networks, never mind the cell, never mind the nucleus or any of that, just by virtue of the mathematics that governs molecular pathways interacting with each other, they can do 6 different kinds of learning. Okay, there are no, you don't need synapses, you don't need you don't need any of that. 6 different kinds of learning, including Pavlovian conditioning, just from it's a free gift from the mathematics of how networks of chemicals turn each other on and off, and we are, and so you can read about that here. We are actually now trying to take advantage of that by training these molecular networks for things like drug conditioning. There are many interesting applications in pharmacology and so on. So what's happening here is that our body is a multiscale collective intelligence. All of us are collective intelligences, not just the bees and the ants and termites and things like that. We are all made of parts. All of our parts are have aspects of cognition all the way down to the molecular level. Already, by the time chemicals are interacting with each other, you have certain kinds of learning taking place. You have causal emergence rising. This is a brand new paper that isn't out yet. It starts very low down on the scale, and every component, every tier of our bodies are able to solve problems. I call this the multiscale competency architecture. Now, when I say that they're intelligent, what do I mean by that? I mean a competency in navigating various spaces. William James defined intelligence as the ability to reach the same goal by different means. So I showed you many examples of that. Plasticity, ways to get to your goals when things are getting very different and everything's changing. Can you still get to your goal? That requires us to talk about spaces and what embodiment really means. So let's talk about this. Because of our evolutionary history, we are kind of obsessed with 3-dimensional space, in which medium-sized objects moving at medium speeds do things that we consider interesting, and we say that's intelligence. So we got birds and maybe even an octopus or a whale or things like that. But the thing is that biology is much broader than this, and the perception action loop that defines intelligent behavior can happen in many other spaces that are hard for us to visualize. And so we have to have a lot of humility here when we look at AIs and various other weird hybrid beings and so on, and we say, well, they're not embodied. This AI is just sitting in a server somewhere, or this organoid is not moving around. It's sitting in a petri dish. It's not embodied. It is embodied. We just are not good at detecting what space it's doing its perception action loop in, because living things navigate transcriptional space, meaning the space of gene expression states. They navigate physiological state space. We're going to talk the most about this anatomical morphospace, the space of possible configurations. So the first thing, as I said, it's very reliable. We navigate morphospace from a point corresponding to an egg to one that's corresponding to something like this. But the reliability or the rise in complexity is not at all why I call it intelligence. The reason I keep calling it intelligence is because of this creative problem-solving capacity, the ability to navigate that space in a way that isn't just moving down a gradient. So the first thing we know is that if you cut early embryos into pieces, you get twins and triplets. You don't get half bodies. The system rapidly adjusts. Some animals, such as this axolotl, can do this in adulthood. So these guys regenerate their limbs, their eyes, their jaws, and many other organs. And if you cut them into, if you cut anywhere here along the length, the cells will rapidly spring into action. They rebuild the limb and then they stop. So this is a classic homeostatic system. You deviate it from its set point. It works really hard to get back there. And then once the error is within tolerance, it stops. So we can call this, this is a kind of anatomical homeostasis. So how could this possibly work? Here's a very simple system where the collective of cells are working on this big project. They all stop when it's completed. How does this group know what to do? How does it know what the goal is? How can it remember what to build? This is a classic problem in developmental and regenerative biology. So we took the approach of looking into and getting inspiration from the one sort of uncontroversial example where we know that groups of cells can store and pursue goals, and that is the nervous system. So in the brain and nervous system, we know that we have a bunch of cells bound into a network. The network, the cells have these ion channels on their membranes that allow cells to acquire electrical states. They can propagate those electrical states through these electrical synapses. All of this is highly regulatable. So it's lots of opportunities for feedback loops and things like this. And that drives a kind of software, the electrophysiology that runs on the brain. And what this electrophysiology does is issue outputs to your muscles to move you through three-dimensional space. And so the story of neuroscience is basically that it arose, the system arose because these electrical networks thought about where you need to be in three-dimensional space and issued commands. And the project of neural decoding is this commitment to the idea that the electrophysiology stores the mental content, that if you were to read and interpret the electrical information, you would know what the animal or human is thinking, what are their preferences, their goals, their plans, and all of that. Okay. So what we know from neuroscience is that the electrophysiology is the cognitive glue of the mind and the brain. It's the thing that makes you more than just a pile of individual neurons. Yeah, it's the thing that holds it together and allows your neural networks to think about things that you don't, to allow you to think about things that your individual neurons don't know. Well, it turns out that evolution figured this out long before nerve and muscle came on the scene. Basically, around the time of bacterial biofilms, all cells have ion channels. Most of them have electrical synapses that they can communicate these states to their neighbors, and every tissue has these kind of electrical patterns. So we asked this question: if your neural networks are thinking about moving you through three-dimensional space, what are your somatic networks thinking about? They're thinking about moving your body configuration in anatomical space. They want to move you in this space that's very hard for us to directly visualize. They want to move you from where you started, which is a single cell, to whatever your correct target morphology is. And all the decisions that they need to make along the way need to be implemented by something. And I'm going to show you how it's implemented, at least in part, by the exact same mechanisms. So bioelectricity is the cognitive glue of the body, exactly how it is in the brain. It underlies the movement through anatomical space. And it's the interface through which we can begin to communicate with that intelligence. In other words, right now I'm communicating with your intelligence using this language interface, but we can modify that and use the bioelectrical interface to talk to the intelligence that is guiding cells to build and then repair and then upkeep your body. So we developed some tools. So in order to do this, you have to build some tools. The first thing we built are ways to read the electrical states of the body, so to listen in to this information. And these are voltage-sensitive fluorescent dyes. So basically, we're reading the voltage states of the cells. These are not models; these are real. So cells, in addition, this is an early frog embryo developing. Now we can see all the electrical conversations that these cells are having with each other. We do a lot of simulation, both tied into the molecular biology that drives expression of these ion channels, to the tissue-wide dynamics, and then eventually things like pattern completion in networks. So which is basically regeneration. So we can read the electrical information. Even more important, we can start to rewrite it. It's not enough to be able to read it. You have to now send signals. We don't use, there are no applied fields, no magnets, no electrodes, no waves, frequencies, nothing like that. We hack the interface that cells are normally using to talk to each other. That is the ion channel. So we can open and close these ion channels that set cellular voltage states. And we can open and close these gap junctions, the electrical synapses that propagate. So we can control the topology of the network, and we control the actual voltage state using all the tools of neuroscience. So ion channel modifying drugs, optogenetics, things like that. So now it's time for me to show you what happens when you communicate with that intelligence. Can we send it signals? Can we what can we do? Well, one thing that we can do is for example, there's a bioelectrical state that, as it turns out, I didn't have time to show you, but as it turns out, there's an electrical pattern, a pattern memory that sets with the position of the craniofacial organs. And what we can do is reproduce some of those patterns elsewhere in the body. So here we injected some RNA encoding a potassium channel. It sets up a voltage state of a pattern of voltage that says to the cells, build an eye here. And sure enough, they build an eye. So this is normally the gut, except we told these cells they should make an eye. And here's what they do. And it's got lens, retina, optic nerve, all that stuff. Now, a couple of amazing things here. The first is that we didn't have to talk to individual cells. We don't know how to micromanage the building of an eye. It's a very complex organ. I have no idea how to do that. What we did find is a very high level prompt that the cells interpret as, we need to build an eye here. So the same way that I'm talking to you now as a collective intelligence made of neurons, the architecture of our brains are amazing, because if I want to give you a piece of information, I don't need to think about reaching in there and tweaking molecular details of your synapses to make sure you understand me and that it sticks as memories. You do that on your own. I'm providing information via a very thin language interface. You control all the biochemistry below to make it happen. That's exactly what happens here. We provide a simple signal. The system, the material takes care of feeding that all the way down to all of the gene expression, the changes and everything else that needs to happen. The other amazing thing is that the material is really smart. This is a cross section of a lens sitting out in the flank of a tadpole somewhere. If we only inject a few cells, so the blue ones are the ones that we actually injected, they decide they need to make an eye, but there's not enough of them. And they know there's not enough of them. And what they end up doing is communicating to the rest of the cells out here. All this other stuff, you don't see the blue signal. We never touched it. It's these cells are convincing these other cells that, hey, you need to help us build an eye. And actually, there's a bidirectional a battle of worldviews going on here, because these cells are saying, let's build an eye. All of these other cells are saying, no, you need to be gut or skin or whatever. And sometimes they win and there is no eye. And so you can actually watch these patterns kind of compete with each other to see who wins. And many other collective intelligences like ants and termites and things also recruit their neighbors when they need to solve a task. So we can communicate novel goals. We can rewrite specific pattern memories. So these are flatworms, and these flatworms are amazing. You can cut them into pieces. Every piece makes a normal worm. We asked the question, how does it know how many heads it's supposed to have? And so we found there's an electrical pattern memory that says one head, one tail. We can rewrite it to say two heads. It's a little messy. The technology is still very, very young. But when you do this and then you cut the animal, you in fact get two-headed animals. This is not Photoshop or AI. These are real animals, and that's because this is the memory that the cells are using to know what does a normal planarian look like. And when you change it, they will build to whatever the goal state is. You can actually rewrite the goal state because it's an agential material. It is not hardwired. And so a single body of a planarian can store at least two different representations of what a correct planarian is supposed to look like. And it's actually a counterfactual memory, because this animal right here that we've changed, until you cut him, he's a perfectly normal, both molecularly and anatomically, he's a perfectly normal one-headed animal. This two-headed state is not true right now. It is a future thing to which you would regenerate if you get cut into the future. In the future, it's a counterfactual kind of time travel, mental time travel, like primitive version of that. So I keep calling it a memory because we found out that if you take these two-headed worms and you cut them later, forever, as far as we can tell, in plain water, no more drugs, no more any interventions, you just keep cutting them, they will continue to make two-headed animals because you have permanently reset what they believe is a correct planarian, not what the animal believes, what the cells believe, right? There are multiple levels of agency here. And it holds. When you change that pattern, it keeps. It's a good memory medium, and you can see the behavior of these two-headed guys here. Not only can you control the number of heads this way, so you can ask the collective intelligence. Let's remember the context here. What we're doing is using the electrical interface to communicate novel goals to the collective intelligence of cells that is making decisions about shape. You can also ask for different shaped heads. In particular, you can ask for heads that belong to other species. So the same hardware, this nice triangular, this species with a triangular head, can make heads that are flat, like this P. felina, or round, like this S. mediterranea, or of course its own normal head. The brain and the distribution of stem cells are exactly like these other animals, between 100 and 150 million years of evolutionary distance. But we introduced no genetic change in the two-headed animals. In these animals, we didn't touch the genome at all. The hardware is completely stock, right? And what's happening is that because of the reprogrammability, because the hardware isn't what drives, it's the decision-making process that drives. The same hardware is perfectly happy to visit other attractors in that anatomical state space that normally belong to other species. In fact, you can get really weird and find attractors that don't belong to any known planarian species that make these weird spiky forms and these cylindrical things and kind of combination things. It's just a different journey in anatomical space. Okay, so the last thing I want to show you is this business of novel goals, because what I've shown you now is that we can modify among the set of goals that animals normally have. So for example, planaria already know how to make a head, and we ask it to build a second head. We call this the electric face, because this is the bioelectric pattern that tells the frog face, here's where the eye is going to go, here's the mouth, the placodes out to the side. So all of these are natural structures, and so we can take this eye pattern and we can reproduce it on the tail of a frog, and the cells will make an eye. But all of these are normal patterns, and if you ask where do they come from, the standard answer is, well, evolution. That basically eons of selection for having a specific anatomy, and everything that didn't have this died out, and now you have this. So these are shaped by evolution. Fine. Okay, but that is a very limiting kind of perspective, because life is much more plastic and much weirder than that, and we know that because of that plasticity, because it is fundamentally an unreliable medium. It is incredibly interoperable, meaning that at every level of organization we can introduce novel materials. Okay, from the subcellular level to the ecosystem level, we can introduce changes. And everything tends to work. These things will always work together. Life finds some kind of a way for it to work together, which means that we as standard modern adult humans are at the center of not only a evolutionary spectrum and a developmental spectrum, where whatever you think is true of our human mind, well, where and how did it slowly and gradually get here? We also have now this additional spectrum where slowly and gradually, by small changes, you can make things that don't look like humans at all, and both technologically and biologically. And of course, this is already here to some extent, but it will be much more prevalent in the future, where you will get augmented and altered humans that are fully taking advantage of their freedom of embodiment to have new senses, new capabilities, to operate in new spaces that traditional humans can't even visualize. And so this kind of thing, where we are trying to say, is it a real human or isn't it? And what are you going to do? Measure whether they have more than 50% of their body or brain modified, or what? It's just not going to work. There's an incredible opportunity and a huge option space for different kinds of beings that were never on the evolutionary tree with us. In other words, they're novel beings. Where do their behaviors and their anatomies come from if it isn't just a genetic history of selection for specific things? So this is now we're getting into the weirdest part of the talk, which is this. Let's ask ourselves, where do patterns come from? I mean, biologists love patterns that come from genetics, and some that come from the environment in the form of physical constraints. But what else? So let's look at this. This is a simple function in complex number z cubed, and so on. If you plot this function using a very simple algorithm called a Halley plot, what you get is something like this. This is the incredible richness that's hiding inside of this tiny little seed. And if you actually slowly change one of these constants, you can make movies like this. These are videos that you can make. It doesn't hurt that it's quite organic looking. You can make all kinds of other patterns. You can find other patterns that are like this. Where does this come from? Okay, where does this particular, why is it this particular picture and not some other picture that is indexed by this seed? Well, there's no story about history. There's no genetics. There's no evolution. There's no lineage that's going to explain why it looks like this. There's also no fact of physics. There is nothing about the laws of physics or the physical universe that defines this. There's nothing you can do in the physical universe to change what this pattern is going to be. It comes from the laws of mathematics. And you can see that the laws of mathematics are actually capable of producing very complex patterns. So could we now find some actual living beings that do not have a history of selection in their embodiment? What would that look like, right? Can we start to understand how novel beings will have new shapes and behaviors? And so I want to introduce you to this. This is something that if I were to show you and not tell you what this is, you would think that it's something we found on the bottom of a pond or a stream somewhere. It looks like a primitive organism. You might guess that it has a genome that's similar to that of a primitive organism. I can tell you that if you were to sequence it, you would find that it's 100% Homo sapiens. These are human cells. These are adult human tracheal epithelial cells that we have put in a process that allows them to reboot their multicellularity. They become this little motile organism. The original donor of those tracheal epithelial cells may be alive or they may not. So even after death, your cells can go on to have a life in a different environment, in a different embodiment. What are the features of this novel organism? They've never existed before. What are their features? Well, first of all, they have 4 distinct behaviors that you could build an ethogram of transition probabilities between them. They have a drastically remodeled transcriptome. In other words, almost half their genome is expressed differently than the cells they come from. Remember, there's no genetic modification here. We don't do any CRISPR, no synthetic biology circuits. These are completely wild type cells exhibiting their creativity in how to be a novel organism when the rest of the body is gone and they're just the tracheal cells by themselves. That is what they do. There's something else they do that's amazing, which is that if you plate a dish of neurons like this and you put a big scratch through it, you put this big wound through it, and you let some Anthrobots run around in that space, they will eventually settle down. They can clump together into the superbot cluster, and what they do is they start to knit together across the gap. Here's what they do: they're fixing the neural wound. So who would have thought that your tracheal epithelial cells that sit quietly in your airway dealing with mucus and particles and air particles and whatnot have the ability to self-organize into a new creature that's never existed before? It doesn't look like any stage of normal human development. It has new gene expression. It has new behaviors, new capabilities. We've only scratched the surface of what these things can do. This is the study of novel beings. And you can do the same thing with frog embryos. So these are called Xenobots. We liberate some prospective skin cells from the top of this frog embryo. They could have died or crawled away from each other or made a 2-dimensional cell culture layer. Instead, they make this thing, and you can see this is one of my favorite videos. Each one of these circles is a single cell. So they've self-assembled into this odd little horse-looking thing that then moves around together as a unit. And then the flash you're going to see, that's a calcium signal. There's some kind of signaling event going on here. So these cells get together and they eventually make this. They make this thing that it can go in circles. It can sort of patrol back and forth. You can make it into weird shapes like these donuts. They have collective behaviors. Here it is navigating a maze. So there's no fluid flow in this maze. It's spontaneous. It moves forward. It takes the corner without bumping into the opposite wall, and then it decides to turn around and go back where it came from. Okay, so it has all kinds of behaviors. One of the most amazing behaviors it has is we call this kinematic self-replication, meaning that if you give it a bunch of loose skin cells, what they'll do, both collectively and individually, is they'll polish them into little piles like this, and the little piles mature to be the next generation of Xenobots. And guess what they do? They do the same thing. And so you get the next generation, the next. Again, to our knowledge, no creature on Earth does kinematic self-replication. there have never been any Xenobots. There's never been selection to be a good Xenobot. Where does this come from? This is what we're interested in understanding and being able to predict. Now, one of the things that we found is that these Xenobots, I'm going to pause this for a minute. One of the things that we found, again, much like the Anthrobots, the Xenobots express tons of genes differently. And among these novel genes was a cluster of sensory perception of sound. And we thought that's weird. I mean, meaning this is normal embryos don't. This is different in expression between Xenobots and normal embryos. So these Xenobots upregulated all this stuff having to do with sensory perception of sound. Why would that be? Could they possibly hear? And so we did this experiment. We basically put a speaker underneath the Xenobot dish, and what you see, oh man, what you see here. Sorry, just. My Keynote quit for some reason. It hates that that one video. Hang on 1 second. Where are we? Sorry, just 1 second. I'll be back. Yeah. So what's happening here is that embryos, Xenobots that don't have the ability to move do not. Man, all right. I'm gonna skip that that slide. It's clearly not going to. Do this. The bottom line though is that basically what I'm showing you is that the Xenobots can actually react to sound. If you play sounds, by their behavior you can see that they can hear. So okay, moving on, just to kind of close up here. Here's the deal. What I've been showing you is that cells during morphogenesis, cells form a collective intelligence that can solve problems in anatomical space. The bioelectric interface, but no doubt other interfaces, are ways that we can use to communicate with them. And one of the things that we are working on is AI, exploiting AI to make tools where we can communicate to the different layers of organization in the body. We're working towards something called an anatomical compiler, where you can draw the animal, plant, organ, biobot, whatever that you want, and the system should give you a set of stimuli that would be applied to cells that get them to build exactly whatever you wanted. Now the key about this is that this is not some kind of 3D printer. This is a communications device. This is really important. We are dealing here with an agential material. We are not trying to micromanage it or to force it. We are trying to communicate with it. And that means that what we really need to do is to crack the interpretation of these various signals by the collective intelligence, very much like neuroscientists are trying to do neural decoding. And so the first thing that we're working on, this is in collaboration with Josh Bongard's lab at UVM, is a robot scientist platform that is going to basically do cycles of making hypotheses, building real physical biobots, observing them, and then learning from that experience, revising its model, and basically try to help us be a translator layer between us and the intelligences we try to control. And so the last just two quick things that I want to tell you is that, you know, I showed you that certain patterns, in particular those Halley plots, and I claim the things that control the form and function of things like Anthrobots and Xenobots are coming from a new source of information that we are not used to thinking about in biology. They are not coming from an evolutionary history. They are not coming from constraints of physics. They are coming from the same place that mathematics comes from, which is a quote unquote Platonic latent space of patterns. People like Plato and Pythagoras have thought about this for a really long time for the facts of mathematics. Where do the facts of mathematics come from? Well, they come from this non-physical space. I'm going to claim that the things that we see in these synthetic models are better thought of as other types of patterns in a space that is organized and has a metric, and that we can systematically explore by creating things like biobots and various hybrid systems and so on, and AIs, of course, and so on. We can systematically explore that space. I don't believe we should be thinking about this as emergence, as just being surprised by facts that just happen to hold in the physical world. I think it's a systematic space that we need to investigate, and this has many implications, because basically, the space of beings that Darwin was thinking about, the natural ones, when he said endless forms most beautiful, is a tiny little corner of all the possible beings that are out there. And increasingly in our lifetime, and certainly in your lifetime, we are going to be confronted with beings that are nowhere on the tree of life with us, because almost any combination of evolved material, engineered material, software, and these ingressing patterns from this mathematical space forms some kind of body and some kind of mind. We are going to have to expand our frameworks for ethics and for relating to other beings, so that we can have a kind of ethical symbiosis with them. And basically the last thing I want to tell you is that this idea of emergent complex problem-solving behaviors that we observe in the world that come from this latent space. It doesn't take life. It doesn't take a brain. It doesn't take a conventional nervous system. It doesn't take any of that to start seeing these things. Extremely minimal things, in particular. And I have to finish up, so I don't have time. But you can ask me in this paper. You'll see that even simple things like short deterministic sorting algorithms, things like bubble sort, have weird, unexpected problem-solving competencies that are not in the algorithm themselves. They do these side quests in addition to sorting the numbers that nobody had noticed in decades of working with these algorithms. And very minimal systems are already capable of doing some things that you would recognize as simple cognitive behaviors. So we are very bad at actually predicting these kinds of things. And just as a reminder, Magritte said to us that our formal models are not the thing itself. This is a representation of a pipe. It's not the pipe itself. And the kind of computationalist formal models that we have, things like Turing machines and whatever, the limitations when people say, well, machines can't do this or that because Turing machines, are limited and so on. Yeah, those are limitations of our formal models. Let's make sure we don't confuse our formal models for the real thing, because nothing is is completely encompassed by the formal model. Not living things, of course, and certainly not things that we think of as machines either. For the exact same reason that the laws of biochemistry don't tell you everything you want to know about a complex mind, the algorithms and the physics of materials does not tell you everything you need to know about even simple mechanisms. Okay, so I'm gonna skip some of this stuff because I don't have time, and just basically point out that the new Garden of Eden that is coming is a lot weirder than the original. And I think we now finally have tools to start understanding and start mapping out this the space of patterns that can be embodied in the physical world as embodied intelligences. And so these are various papers. I'm happy to send these out if anybody wants to dig into this stuff. Most importantly, I just want to thank all the people who did the work. So here are the postdocs and the scientists that did the things I showed you today. Lots of collaborators. Our funders' disclosures. So there are 3 companies that are funding some of our work, and always have to give the most credit to the model systems. They're the ones who do all the hard work. So thank you so much, and I'll take questions.